Hello everyone, welcome back to our channel. I'm Dr. Ashmita. In this video, we'll continue with the series of G protein coupled receptors. In our last video, we have discussed in detail about the G stimulatory, G inhibitory, and the GQ pathways for the GPCR signaling. In this video, we'll continue with the G1213. So let's have a quick recall. Whenever there's a ligand binding, the signal is transmitted to this G protein complex. It is first received by the alpha subunit, which undergoes this conformational change and making it active. That active G alpha will separate out from this beta gamma dimer, and the path chosen, either stimulatory, inhibitory, or this Q pathway or 1213 pathway. Which pathway it will choose, it depends upon the type of ligand and receptor being activated. So in this video, we'll study this G alpha 1213. So let's see what are the receptors and ligands responsible for this G1213 pathway. Firstly, there is angiotensin 2 receptor acted upon by angiotensin 2, and this thromboxane A2 receptor acted upon by thromboxane A2. Thirdly, oxytocin receptor, ligand is oxytocin. Fourth is endothelin receptor, the ligand being endothelin 1. And fifthly, M3 muscarinic receptors, the ligand here is acetylcholine. These are the few examples of the receptors and ligands, GPCR receptors and the ligands that choose this G1213 pathway. So let's understand in detail this 1213 pathway. So upon receiving the signal, the G alpha subunit got activated. It separated out from this beta gamma dimer. Now this particularly G12 and 13 got activated. It will directly bind to the RhoGEFs. RhoGEFs are the Rho guanine nucleotide exchange factors. These are present on the inner surface of plasma membrane. So this is a cell membrane. On the inner surface of the cell membrane, this RhoGEFs, the Rho guanine nucleotide exchange factors are present. So the G alpha, that is a 1213, will directly bind to this RhoGEFs. So what are these RhoGEFs? Three examples are P115 RhoGEF, LARG, thirdly PDZ RhoGEFs. These are the few examples of the GEFs. Now I would like to draw a diagram. This is a plasma membrane. On the inner surface of plasma membrane, I have drawn this RhoGEF. This is a RhoGEF. The activated, this is the active alpha 1213. It will directly bind to this. Because of this binding, there is a conformational change in this RhoGEF. Conformational change is the change in three-dimensional structure of this RhoGEF, because of which there is a DH domain that gets exposed. So here is the DH domain. It is getting exposed. The DH domain is present on the RhoGEF. The DH domain on RhoGEF gets exposed, because of which RhoA gets recruited. RhoA is present. RhoA is a protein present in the cytoplasm. It is bound to GDP in inactive state. Whenever the DH domain is exposed, the RhoA is pulled towards this RhoGEF because of DH domain, and now it tightly binds to this DH domain of the RhoGEF. So now the RhoA, which was inactive, like it is bound to GDP, now it becomes active by binding to GTP. This RhoGEF will push the GDP out. It will push the GDP out and it will insert a GTP. So it is inserting a GTP. Now it is said to be an active RhoA. It is an active RhoA here. So this active RhoA, now it will bind to effectors. So all this is happening at this surface level, at the plasma membrane level. This activated alpha binding the RhoGEF present on the inner surface of plasma membrane. Because of binding, this DH domain got exposed, because of which this RhoA present in the cytoplasm gets pulled towards RhoGEF at the membrane. And because of this binding, this GDP will be exchanged for GTP. Hence, it becomes active Rho. Now, this active Rho will act upon the effectors. It will bind to effectors. What are the effectors? Here it is called a ROCK effector. ROCK means Rho associated coiled kinase. These are the Rho associated coiled kinase present in the cytoplasm. They will act on this effectors. Now this activated Rho will act on the effectors. The effectors here are the ROCK, which has two isoforms 1 and 2. ROCK 1, ROCK 2 are the two isoforms of this kinase. And other receptor are, this is 1, this is 2, MDR. This is the other receptor. Other effector. And this effectors, like upon getting activated, for example, this is the activated ROCK 1, 2. Now the final effect is produced by this activated ROCK. It will phosphorylate the targets. The targets here are myosin light chain MLC, which cause contractility, LIM kinase, which will stabilize the actin filaments, and proteins promoting the focal adhesion maturation. These are the few targets upon which the activated ROCK will phosphorylate, producing the final effect. The final effect is contractility, contraction. It will cause cell contraction. So what does all this mean? So this ROCK, the final effective ROCK is it will act on the actin myosin cytoskeleton, actin and myosin cytoskeleton, and finally it will cause cell contraction. It will result in contraction. It will increase the contraction. This is the final end result given by this activated ROCK in this pathway. So let's see a few other examples where this ROCK pathway gets activated and with clinical significance. Firstly, in the vascular smooth muscle, the RhoA-ROCK, it will phosphorylate MLC, that is the myosin light chain. It gets activated. If myosin light chain gets activated, the end result is contraction. And also, it will inhibit MLC phosphatases. MLC phosphatase means it will remove phosphate from this MLC. If you remove, it will become inactive. So you inhibit that phosphatase. Either way, you are maintaining this contraction. The end result is to produce contraction. So it produces long-lasting vascular tone because of the smooth muscles of this vascular channels. When they get contracted, it will maintain this vascular tone of the vessels, and it helps maintain the blood pressure. Because of vascular tone, the blood pressure is maintained and the vessel diameter is maintained. So the receptors here, example of receptors and ligands in this vascular smooth muscles are angiotensin 2 receptor acted upon by angiotensin 2 and endothelin receptor acted upon by endothelin 1. This is about the vascular smooth muscle cells. Coming to the airway smooth muscle, in airway smooth muscle cells, this ROCK will increase calcium sensitivity. Because of increased calcium sensitivity, there is tightening of the cells. The smooth muscles get tightened, which is actually a protective function. It will protect the lungs by narrowing the bronchi during irritant exposure. Whenever there is irritant exposure or exposure to cold air or any pathogen entry, so it will have a protective function by constricting, by like narrowing the bronchi, by tightening the smooth muscle cells of the bronchi because of calcium sensitivity. The receptor, example of receptor here is M3 muscarinic receptors. M3 muscarinic receptors. The third example here is again a smooth muscle of the GIT or the gastrointestinal tract. This ROCK will mediate tonic contractions in the esophagus, stomach and intestine and help in peristalsis, the peristaltic movement. That is, the food passes through the food pipe with a peristaltic movement. That is like a synchronized contraction. It will maintain the sphincter tone, that is, the lower esophageal sphincter and the ileocecal valve sphincters. These sphincters are maintained, and it will help in digestion of the food. Fourth example is platelets. They help changing the shape and activation during clotting. Here the ligand is thrombin. Thrombin activates PAR receptor wherein the activated ROCK will cause stress fiber formation. Stress fiber formation are this actin and myosin bundles, actin-myosin bundles of the cell getting tensed and help in contraction. So rapid shape change and clot stabilization. The ROCK causes these three effects that will result in platelets will change their shape within seconds to form a plug. Therefore, it will help in this clotting. And fifth example here is fibroblast. In the fibroblast, this RhoA and ROCK will activate the fibroblast. Activated fibroblast will behave like a myofibroblast. It will transform into a myofibroblast, means it will have contractile function. So they produce stress fibers. Again, the actin-myosin bundles getting tensed, and there's collagen deposition and wound contraction because of this myofibroblast means it will have myo means contractile function. So it will help in wound healing and tissue repair. In the kidney, it will it will have an effect on podocytes. The RhoA and ROCK will control the actin in the podocyte food processes. So consider this as a filtration barrier. This is the basement membrane. These are the endothelial cells. This is the food process of podocytes. So this gap is the slit diaphragm. So it will stabilize this slit diaphragm by it will controlling this actin present in this podocyte. It will control the actins of the podocytes and maintain this because of this contraction or relaxation of the podocytes. It will maintain the slit diaphragm. So it will prevent proteinuria. It will prevent leakage of proteins through here. From the capillaries, it will prevent the leakage of proteins. This is about the effect in kidneys. Coming to the leukocyte migration, that means it has a role in immune response. The RhoA and ROCK will contract the rear part of migrating leukocytes. Consider this as a cell. This is a front edge. This is the back edge. That is the rear edge. So this back edge will contract. That means the cell is propelled forwards. It will be propelled forwards. That means it will help in migration. Therefore, the cells will squeeze through the endothelial gaps. When it is contracting from behind, the cell is pushed forward and it is squeezed through this endothelial gaps. It is like squeezed through the endothelial gaps. So leukocytes will move towards the site of injury. So this is about the leukocyte migration. What happens if this pathway is overdone, if the signal is not terminated, and if the signal continues and all this activate this RhoA ROCK pathway like it is overly activated, what will happen? What is the pathology? So in vascular smooth muscle cells, as we said, normally it will help maintain BP in vessel diameter and vascular tone. If it is overly done, it will lead to hypertension. And in airways also, like it will have protective narrowing, but if it's overdone, it will lead to asthma. In the gastrointestinal system, it will aid in digestion. If it's overdone, it will result in inflammatory bowel syndromes, functional constipation, achalasia cardia, and the gastroesophageal reflux disease. In the platelets, if it's overdone, if the pathway is over, then it will lead to excess of clotting. Normally, it will help in formation of plug. If it's excessive, then it will lead to excessive clotting and will result in deep vein thrombosis, deep vein thrombosis, myocardial infarction, and stroke. In the fibroblast, normally it helps in wound healing and tissue repair. If it is like overly done, because of the myofibroblast overly working, it will overly contract. It will result in hypertrophic scarring and keloid. It and excess collagen deposition will result in liver cirrhosis, pulmonary fibrosis. And kidney podocytes, it will prevent proteinuria. But when it's like defective, it will result in diabetic nephropathy, focal segmental glomerulosclerosis, and hypertensive nephrosclerosis. Whereas in leukocyte migration, they'll help the leukocytes migrate towards the site of injury. If it's defective, it will result in arthritis and other autoimmune disorders. And also, it has a role in invasion and metastasis of cancer. The cancer cells will hijack this rock to gain the motility and resistance through apoptosis. So it will gain the motility means it will easily invade. It will invade through the adjacent stroma. It will invade through the basement membrane, breach the basement membrane, and like travel to distant places. And also, it is resistant to apoptosis. Therefore, even it will exploit this ROCK pathway. The tumor cells exploit the ROCK pathway to invade and metastasize. This is about the clinical relevance of this G1213 pathway, that is the RhoA and ROCK pathway. So with this we have completed the G protein couple receptor signaling. This is the last part of it. We have studied the stimulatory, the inhibitory and the Q part in our previous videos. Please go back to it. And if you find this useful, please like, share and subscribe to our channel. Thank you.